Hi everyone, I'm Nicole Gasdik, and I will be talking about ion torrent. So what is ion torrent? Ion torrent is a DNA sequencing technique based on the detection of hydrogen ions released into growing DNA templates during the incorporation of new nucleotides. When the hydrogen ions are released, there is a pH change, which makes a clear correlation between chemical and digital events. The bases are called without scanning, cameras, or light. No modified nucleotides or fluorescence is used in ion torrent. So there's 6 steps in ion torrent. Template preparation. The first step in ion torrent sequencing involves preparing a DNA sample that you will want to sequence. This sample can be genomic DNA, cDNA, or any other type of DNA that you're interested in sequencing. Emulsion PCR. The prepared DNA sample is then used to create many copies of the DNA fragments in tiny water and oil droplets, known as microreactions. This process is called emulsion PCR and involves attaching DNA fragments to beads within these droplets. Sequencing by synthesis. Each bead with an attached DNA fragment is placed in a well of semiconductor chip, which is specifically designed for ion torrent sequencing. The chip contains millions of individual wells, and each well holds a single bead. Sequencing process. As the sequencing process begins, a nucleotide base are added one at a time to the DNA template. When complementary bases are added to the template, it releases a hydrogen ion as a byproduct. The release of these hydrogen ions is detected by the semiconductor chip. Signal detection. The semiconductor chip records the release of hydrogen ions from each well as DNA sequencing reaction proceeds. The rate of ion release is directly proportional to the rate of nucleotide incorporation. The signal is then translated into DNA sequence for information. Data analysis. After the sequencing run is complete, the data is processed using specialized software to convert the detected signals into DNA sequences. The software aligns the sequences and generates a consensus sequence, which represents the original DNA template. So in figure A, this is a cross-section of the sensor array. This shows all of the pH sensors and the design of the semiconductor. In figure B, this is showing the dips for the beads to fit into. So what happens is your solution with the beads goes over the plate, and the beads fit. One bead fits into each of these wells first, and then you put on your solution with your bases in it. And if the base can bind to the bead, it will, and it will give off a hydrogen ion like in C with the growing chain, and it giving off a hydrogen ion, and it will give off a voltage from the pH. And from that voltage, different bases will give off different voltage amounts. You'll use that and put it through the DNA sequencing for your data analyzing. So what are the major advantages of using ion torrent? One major benefit of the system is the rapid sequencing speed compared to fluorescence-based systems. Another benefit is the low cost to use the system. In this photo taken from a paper, They compared ion torrent sensitivity and real-time CT values compared to the MySeq, and the sensitivity was just about the same as the MySeq, and the real-time CT values, um, they were close to the MySeq with some outliers. But if you were planning on using the ion torrent compared to MySeq, you would have basically on par with MySeq with a lower cost and faster turnaround. The system also has disadvantages. The length of reads is only between 200 and 400 base pairs. Synchronous sequencing will give you much longer reads if you decide to use that route. If there are multiple of the same nucleotide in a row, it becomes difficult to tell the pH electrical change because it's not a one-to-one electrical change. It's very difficult to tell if there are 5 of the same nucleotides in a row or 6, for example. The error rate is much higher in ion torrent than in other next generation sequencers. In this example, in figure A, MySeq was compared against ion torrent in reverse and ion torrent forward. The ion torrent had about 1.5 error frequency in both the reverse and forward in 100 bases, where the MySeq had about 0.9 error frequency in 100 bases. So this figure comes from a paper back in 2020 about coronavirus and how someone self-diagnosed himself as having coronavirus, and they went to the hospital, and what these doctors did is they did the routine coronavirus test where they did a nasal pharyngeal swab, they did RNA extraction, they did rRNA depletion, and then they library prepped it and RNA sequenced it. They had suspected that it would be coronavirus, so they took these steps to next generation sequence it to make sure that that was what it was. Then they went through and did a quality filter. They removed the host reads, so any human reads. They did a config assembly, so anything that was partially together, they put everything together. They did a sequence similarity search in BLAST, and they sent it out to another company. And they did the config extension and redundancy removal. In B, this is when they got their sequence similarity back, and anything in red under assembled configs is high likelihood that it is similar between the reference genome and the config that they had sent out. So as you can see, there's about 3 spots that had very high likelihood that they were the same. So they were pretty sure that it was coronavirus that they were looking at because their reference genome was coronavirus.